The cell cycle. We all know it. It goes interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. But how does it work? G1 is the first phase, part of interphase, and the longest period. This is the resting period where the cell grows. At the end of the stage, there's a checkpoint which controls whether the cell will divide, delay, or enter a resting stage. If it's approved, the cell enters the synthesis phase, where it replicates its DNA. Once the cell reaches the G2 phase, the cell is prepared for division. If it's approved again, the cell reaches the mitosis phase, where prophase, metaphase, anaphase, and telophase occur. By the end, the cell reaches cytokinesis, and if things go well, it divides, creating a new daughter cell. In order for things to go well, there has to be an even number of tumor suppressor and proto-oncogenes. Tumor suppressor genes, or TS genes, inhibit cell division, acting like brakes. Proto-oncogenes encourage cell division. The problem with this can arise if one of these genes becomes mutated. Mutations are cancer-causing agents that cause changes in your DNA. Mutations can occur from miscopying, environmental factors, and family history. Environmental factors are things like UV exposure, radiation exposure, coal tar exposure, etc. It is common, though, for someone to not have been exposed to any of these things and still get cancer. There are precautions you can take, but there is no proven method of cancer prevention. Once one of these genes have become mutated, one of these two things can happen. When proto-oncogenes become mutated, they become oncogenes, which stimulate excessive division. When tumor suppressor genes become mutated, they become inactive. The key molecules that control cell division are called CDK and cyclin. Normal cells divide after receiving the signal from these chemical messengers. Unfortunately, cancer cells do not obey this rule. Cancer cells will divide even if they do not receive the appropriate signals and can divide indefinitely. Eventually, there will be a mass of cells created which can turn into a tumor once large enough. At first, it will take a very long time to create a mass large enough to actually become visible. A mass of cells could be growing for 10 years and only be the size of a pinhead. The tumor experiences 4 mutations before becoming more invasive and actually breaking through the membrane, which can take a very long time. This tumor cannot be detected. Once the tumor has become large enough, it can use the blood vessels as highways and can travel to new locations all around the body. This is the tumor metastasizing. If the tumor or the metastases are left untreated, it will grow and interfere with the functioning of the body, which eventually leads to death. Over the years, many treatments for cancer have developed. One of the most unique and effective treatments is called endostatin. It's different from other cancer treatments in that it doesn't poison cells. Instead, normal cells are targeted. The goal of this treatment is to starve cancer by cutting off its food supply. Dr. Judah Folkman was a laughingstock when he made it public that he believed tumors could not grow larger than a pinhead without a blood supply. Dr. Folkman became interested in finding a molecule that could inhibit blood vessel growth. One drug he found that could help treat people with multiple myeloma is called thalidomide, which was originally used to ease morning sickness for pregnant women. However, this drug caused birth defects in babies. For morning sickness, this drug was not what you wanted, but Judah Folkman found use for it in treating multiple myeloma, and it worked. He found another treatment as well, called endostatin, named from angio, which means blood vessel, and statin, which means stop. For the mice that he gave endostatin, their lungs were clear and had not metastasized. They didn't have lung cancer. Anti-angiogenesis drugs can treat more than cancer, however, such as arthritis, psoriasis, and macular degeneration.